Thank you to the organizers for the opportunity to present our novel, non-invasive smart sticker technology and the Derm Tech approach to precision dermatology. As a brief introduction, Derm Tech is located in La Jolla, California, and has a CLIA-approved laboratory for the processing and commercial analysis of the smart sticker. Derm Tech additionally holds broad patents on the isolation of genomic and proteomic material from the smart sticker. In 2015, Derm Tech launched an approved diagnostic test for the identification of melanoma, which we will discuss in a few slides. Finally, Derm Tech has a growing pipeline of products, partnerships, and collaborations leveraging the non-invasive smart sticker for skin cancers and inflammatory skin diseases. On the left side of your screen is an actual smart sticker following the non-invasive collection of a skin sample. Once collected, you can clearly see the layer of keratinocytes attached to the smart sticker. Due to the biological nature of skin, keratinocytes containing genomic and proteomic information migrate from the basal keratinocyte layer towards the outer layer of the skin, also known as the stratum corneum or the cornified envelope. While initially believed to be dead cells, the keratinocytes in the stratum corneum have largely retained their genomic and proteomic properties and therefore can serve as a non-invasive surrogate for a skin biopsy. Non-invasively sampling the skin using 4 smart stickers collects approximately 1.5 milligrams of material from the entire lesion for further genomic and proteomic analysis. In many cases, genomic and proteomic material isolated from the smart stickers can identify changes in the skin before those epidermal changes are visible to the human eye. This makes the smart sticker an ideal approach to both precision and personalized dermatology. Finally, the proprietary properties of the smart sticker preserve genomic and proteomic material in the skin for up to 10 days at room temperature and greater than 1 year when stored at -80 degrees. Dermatology offers a unique opportunity to directly assess the diseased tissue. However, visual assessments are largely subjective, and clinical studies have shown a high degree of discordance between investigators or physicians. Additionally, in challenging times like this past year, it may be difficult for patients to schedule in-person visits with their dermatologist for a skin assessment. The combination of technological advances and improved understanding of human disease has given rise to a new era of precision and personalized medicine. As a pioneer in dermatology, the smart sticker has the opportunity to transform medicine by non-invasively evaluating the heterogeneity of larger populations and stratifying patients based on genomic and proteomic profiles, diagnosing patients with a particular dermatological disease by using genetics and proteomics to identify biomarkers of disease and subgroups of disease, and collecting inflammatory signatures prior to and following treatments in order to predict and track responses to therapeutic intervention. Approximately 200,000 people are diagnosed with melanoma each year, which accounts for more than 7,000 deaths. Studies have shown that early detection of melanoma and corresponding action is critical to improving clinical outcomes, suggesting that early detection may have prevented many of those deaths. The Pigmented Lesion Assay, or PLA, is a non-invasive genomic assay that uses the smart sticker to collect skin samples from suspicious moles or lesions without the need for surgical biopsy. Following collection, genomic material is isolated from the smart sticker at the Derm Tech Gene Lab and then analyzed for the expression of link and PRAME. Long intergenic non-coding RNA 518 is a novel gene identified by Derm Tech that plays a role in melanoma proliferation, invasion, and is detected in 84% of histopathologically confirmed melanomas. PRAME, or preferentially expressed antigen in melanoma, is an established melanoma gene that promotes tumor progression and is associated with 83% of histopathologically confirmed melanomas. Overall, 93% of confirmed melanomas express either link or PRAME. In addition to link and PRAME, telomerase reverse transcriptase, or TERT, promoter mutations have been identified in 73% of histopathologically confirmed melanomas. TERT has previously been shown to be associated with invasive and aggressive melanoma and is associated also with poor survival. Including the mutational analysis of TERT to the PLA has increased the sensitivity of the PLA to 97% and is associated with a negative predictive value of 99%. Overall, the PLA test is an essential tool for the early detection of melanoma, which is associated with improved outcomes and has eliminated more than 90% of unnecessary biopsies, which were ultimately determined to be not to be melanoma. In addition to melanoma, the research and development team at DermTech is evaluating additional rule-out tests for non-melanoma skin cancers, cutaneous T-cell lymphoma, and inflammatory skin diseases. Inflammatory skin diseases affect millions of people throughout the world and are associated with an impaired quality of life. As an example, psoriasis is a chronic autoimmune skin disease characterized by significant Th17-mediated inflammation. However, there is heterogeneity amongst the patients with psoriasis, as not all patients respond equally to treatment. Using the SMART sticker, we collected samples from healthy controls with no history of skin disease and patients with active plaque psoriasis. Genomic material was then analyzed for the expression of 13 genes known to drive disease pathogenesis. The heat map on the left side of your screen illustrates the differences between healthy control skin on the far left, lesional and non-lesional psoriatic skin, indicated as NL and PSOR. Based on the initial analysis, we further investigated whether there were subgroups within psoriasis. Interestingly, further interrogation of the lesional skin revealed subgroups of psoriasis patients with differential expression of inflammatory cytokines and Th17 activation. While this pilot analyzed a small number of patients, it suggests phenotypic differences could influence clinical responses to therapeutic intervention. Atopic dermatitis is another chronic inflammatory skin disease. This one characterized by intense pruritus and increased Th2 and Th22 signaling. In recent years, several therapies have targeted the Th2 axis by blocking either IL-4 receptor alpha directly or blocking IL-13's engagement with the receptor. While these therapies provide a therapeutic benefit, less than half of the patients achieve a clear or almost clear, suggesting additional inflammatory pathways play a role as well. Using the SMART sticker, we evaluated the expression of key inflammatory genes in 31 moderate to severe atopic dermatitis patients. The graph on the right side of the screen indicates the percentage of atopic dermatitis patients which expressed a particular gene. As expected, CCL17 or TARC, which is a biomarker of disease severity, was expressed in almost all of the patients. Similarly, IL-4 receptor alpha and IL-13 receptor alpha 1, which are expressed on a majority of inflammatory cells, were expressed in the skin of every single patient. IL-13 has previously been highlighted in publications as the key component for Th2 signaling. So it was very interesting that we observed IL-13 expression in 17 out of 31, or only 56% of patients in this study. While preliminary, this may explain the current response rates for IL-4 and IL-13 targeting therapies. Additionally, IL-22 was expressed in approximately 50% of AD patients, corresponding with the percentage of patients experiencing significant therapeutic benefit when those pathways were targeted with an anti-IL-22 monoclonal antibody. Overall, this data suggests the value of phenotypic and genotypic stratification of atopic dermatitis patients for emerging therapies. Currently, more than 3,000 skin diseases have been identified and described. In some cases, standard procedures may not be sufficient to properly diagnose patients during the first visit. This increases the overall burden of skin disease and can dramatically impair the quality of life for patients until they receive proper care. Integration of the SMART sticker in non-invasive, precision and personalized dermatology approaches has the potential to significantly improve the lives of millions by sampling the entire lesion, collecting skin material for subsequent genomic and proteomic analysis, and driving diagnostic approaches for skin cancers and inflammatory skin diseases. Thank you for your attention. I encourage you to visit dermtech.com to learn more about our innovative platform and approach to precision dermatology.